Chapter 12 is really cool. It's on biosignaling, on the cell signaling pathways. Sometimes these are called cascades. And unfortunately, I do believe that this is really the only class where we really get to go into it, because unless they really change cell-molecular, they just didn't have time. You can't cover everything possible for cell biology in one semester. And so you probably don't get to go through the cascades and signal cascades in very much detail. And so here we're going to talk about the six general classes of the biosignaling pathways. There we go. So first up, we have what's called signal transduction, which is actually a whole field of cell biology, and it's used a lot. And it's a universal property in all living cells. Okay. And so what do you suppose? Like, what do they mean? It's kind of like a buzzword they like to say, or a buzz term. I guess technically it's two words. What is signal transduction?
Some kind of signal comes from outside the cell and it causes like an intercellular change.
That's right. And so many times, like, yeah. And so what would be one example? Okay. How do you mean?
So like growth hormone would be transported to the cell, binds to a receptor on the cell, which would cause part of the G protein to go to the nucleus to increase transcription or something?
Close, but yes, you're getting the binding of this on the outside in order to elicit the increase in the transcription, so you can have a division in the cell growth on the inside. There's going to be multiple parts to that as we go along, but just in general, which when we talk about transduction, many times we're talking about even a change from one. Well, we'll get into that in a moment. But so you have some type of information, you get the cellular response, and it results in a chemical change or even a morphological change there. And there are 4 parts to all signal transduction. Okay, I don't know if you have like some type of mnemonic device that you want to memorize this, or SAD-I is okay, or SADI or whatever, but it's specificity, amplification, desensitization, or adaptation, and integration. So that's why there are the 4 stages. Right? And so whenever we say specificity, what do you suppose that what does that mean without saying any type of form of the word specific?
It is unique to that.
Oh, good one. Okay. So, yes, we're talking about some type of unique property there. In particular, we're discussing let me make it to where I can get it all on here. I don't know why it makes it so large whenever I first import it. But specificities only have 2 components to it. There's an affinity for it, and there's cooperativity. Okay. And so we're talking about an affinity for the binding. Almost always, there is one exception I can think of. Well, technically there's 2 exceptions, one big one and one kind of like subclass that are the exceptions to this rule. But almost always we're talking about an extracellular event of binding that's going to cause and elicit some type of internal change. There's one that actually has to go inside before it causes a change, and then there's another type, the subclass, where it's all internal. There's actually it's not an external event at all. But we have to have some type of affinity for whatever it is, and then there's a cooperativity between the specificity, or that binding event, with whatever is going to be causing the change within the cell itself. And so this is the picture that your book shows. The fact is a signal molecule will fit directly on its binding site due to the complementarity of the receptor, so that other signals do not fit. Of course, now there are some terms like agonist and antagonist, especially with respect to medicine when we talk about medicinal chemistry. Whereas, just to review, what would an agonist do when we're talking here about with respect to specificity on a receptor? An agonist.
Increase the Correct.
And so an agonist binds to that binding site and will elicit the same response or even possibly an increase to it. So it has something similar. Whereas an antagonist would do what? It's going to decrease or turn it off. Okay, and so that's why an antihistamine binds to the histamine receptor, but it doesn't elicit that response. Or it can bind to a different location on the receptor to where the signal molecule can't. And so, but once again, those are just some terms in case you're reading anything in the literature with that respect. But when we're talking about specificity here, we're talking about the binding of a specific signal or a unique signal molecule on a particular binding site, and there's just complementarity to it. Okay.
So the antagonist creates no effect, not like the opposite effect.
That's correct.
Okay.
Well, because usually when we say an opposite effect, it's really just turning something off. Because instead of it being a light switch on and off, many times we're seeing with these cascades that it's more of a fine tuning. And so, because there's lots of redundancy with some of these. For example, with insulin, it's a bifurcated system for the insulin pathway. Stage was amplification. So what does amplification indicate or mean?
Turn up or turn down.
To turn up or to turn down? Do you amplify something by turning it down?
Turn up.
And turn up makes me think of what?
Turned up.
Well, I'm going to say makes me think of turn ups.
Oh.
Oh. Generational
Let's go with yours.
So, yes.
We gotta show you a music video later.
Oh, we've already been talking about music videos in forensic science, so. Oh my goodness. But yes, we're talking about amplification right now. And so how essentially a single binding event or single signal will be amplified, much like this phonograph right here. Hence the term amplification. So this figure from your book is just to indicate what I meant by that. So you have a single signal, and that, which I don't know if anyone's ever explained this to you, and I don't want to just assume, but this symbol right here, was that green triangle? That means activate, okay? Whereas if it's red circle with an X in it, that would mean to feedback, or means inhibit, okay? And so this signal usually will activate one enzyme, and this enzyme will go to the next tier. It's sort of like a pyramid scheme. It's biochemistry pyramid scheme. Next tier of enzymes, and this next tier of enzymes go on, and they have other effectors that they affect. And so you have a third tier of enzymes which will go on and do their effects and possibly go to a next tier. And so it's this, that's hence the reason why it's called a cascade. So it is exponential. It's not a linear process here, which many of you who've seen in papers or some of your other classes may have seen how, for example, one thing like insulin can have so many different effects. And one of the reasons why is it's a bifurcated cascade, and so part of it goes on through what's called a part of it goes on through a protein kinase, and then part of it goes through an inositol signaling cascade. Okay, so we have desensitization. Some people also refer to adaptation. And so if we didn't have some type of desensitization, it would always be in the on or off position. Okay, so what happens here is much like the threshold with respect to a nerve signal, is if the stimulus falls, or any time that it falls below that threshold, your receptor will start to become sensitive once again. So it's not just always on. Whereas adaptation is just a way that it can be modified later. And one example of that is that feedback inhibition, where a product or a metabolite further down the process comes back and will down-regulate the receptor, or could possibly even turn it off. And once again, one thing that people forget about is they just think if something binds, it's turned on and it's just on. or it's turned off and it doesn't come back on. But you have to realize that all of this, or almost all of these will have some type of KD. So whenever the concentration falls below a certain amount, then it's less likely to bind. Or if it's above a certain amount, it's more likely to bind, since it's in equilibrium. It's just a dynamic process. As I was talking about, so in this one we see the little green triangle versus the red, X. And you'll see lots and lots of these next semester whenever we start talking about the metabolic pathways, and they'll show how certain things will be effectors either downstream or upstream of where they, of where the actual enzyme that gets affected is at. Okay. Which I liked this figure. It's calculus integration. It's a faith integration. So what do we mean by integration in this sense, and not in calculus integration? Even though some of you may be able to do some of this from your calculus classes or studying for the DiffEQ exam. So what is integration biologically? What are we meaning by that? Anyone?
Adding it into a process.
Okay, I can take that. And so, am I going to have to do what I do with, organic chemistry and start to get out and, that student that was just in my office, he was excited. He got candy. So, there you go. Okay, you're welcome. He asked me, can I have one of each? And I was like, sure. And he's like, well, I don't really care for Laffy Taffy or the Gobstoppers. He's like, oh, well, then you don't have to have one. That, and to my knowledge, I don't have any Laffy Taffy, so not an issue. All right. But here, integration is actually, many times we're talking about two different signals that have opposite effects. Okay? And you can almost picture it as being a way to fine-tune, whoops, you get this, way to fine-tune the process. And so this change, which actually technically would be more of a, derivative than an integral, but beside the point. It's this change in the concentration of this metabolite or this intermediate that can actually have differing effects. Okay. And so you can also have multiple receptors that can affect or be integrated into the system to either fine-tune it. In fact, one example that we'll talk about, I think it's in the I think it's not until the beginning next semester, where it has 6 different effectors. And so it can really, that enzyme system can be very, very fine-tuned. And so, once again, it's not like this kind of on and off, but it'd be like the little dial, which we don't have in here. So what are some of the forms of stimuli that we could have? Like, what's one type of stimulus? You can have some type of electronic. And when we say electronic, what do you mean by electronic? Okay. What else? What's stimulating you right now?
Capture.
Caffeine. And so what?
Your lecture.
Wow. And so what sense is it? Okay, that's what the Gobstopper too. Okay. What sense is it that's stimulating about this lecture? Is right. We're talking about hearing. You could have a visual, which would be light. There's lots of that. We have taste. It's gustatory. Because I was looking at that big bowl behind the ray. Like that's like mammoth. And so. And so we could have taste. We could go on and on and on with all these different types of stimuli that would be possible here. But that is huge. It's like you could feed like a entire family out of that thing.
You're welcome.
Thanks. All right. So here's just a list of some, and as we mentioned, we've talked about, and I really like some of the wording on these, such as saying gustatory or taste, it's a tastant, smells and odorant, and of course we have pheromones, growth factors, the hormones. The extracellular components, we're talking like integrins and things like that. Antigens, which I'm surprised those of you in immunology didn't mention that. And then of course we have like mechanicals. And those can go on and on and on. Lots of different types of stimuli, types of signal transducers. Once again, what's a transducer? That's like a great term, but what is a transducer?
Transmits a signal.
It does what with the signal? Okay. Well, you're kind of getting all. Many times we're talking about something that can actually take a signal and not only conduct it, but also change it from one form to another. So we can have like a physical event get a chemical response or something like that. Okay. But there are 6 broad classes, and almost everything will fit in at least one of these classes. The first class, and one that we'll go into most detail, is called the GPCRs, which stands for G protein coupled receptors. The second largest class are the RTKs, which is receptor tyrosine kinases. Right? And knowing what you know about just the names, just to review, what does a kinase do? Besides being an enzyme? The ACE part is an enzyme, but what does a kinase do? Adds or removes a phosphate using what? ATP or GTP. And knowing that, what do you suppose a tyrosine kinase is specifically going to do?
Tyrosine.
And so what's going to be phosphorylated? A tyrosine, okay? So ATP or GTP, a phosphate will be removed from an ATP or GTP and added to a tyrosine. That's what's on all the RTKs. And then there's the RGCs, which is receptor guanylyl cyclase. And most of those are extracellular. We're going to find out about one subclass that can actually be intracellular. Intracellular. Then we have the integrins, the adhesion receptors, the gated ion channels, which is the one that you probably should already know the most about, because those are what you talk a lot about that in biology and anatomy, physiology, and things like that. And then we have the nuclear receptors, which is a class that's very unique because it is the only one that's has no extracellular component. Because the receptor is where does the receptor go? It's in the nucleus. And so.
On the opposite side of the membrane, conducts the message.
In a way, yes. I think you're even overthinking this. So for a second messenger, so remember, every GPCR must have 3 things. It has to have the integral effector protein, essentially the receptor, which must have an intracellular and effect an intracellular secondary messenger, and then must have a G protein. Okay, but what does it mean? A second or secondary messenger means, first of all, it means it is not the first, so it would become the second in the loop. Okay, and so some of the examples in calcium, which is the one that you've talked about before with respect to like muscle movements and things like that. Well, I've mentioned inositols in the past, so inositol triphosphate, and there are also other inositols. Diacylglycerol, which where does that come from? What is diacylglycerol?
Isn't it just a sugar that's been modified?
Close. Not quite, but first of all, instead of the acyl part would be the fatty acid, and glycerol is the glycerol backbone. And so we haven't spoken specifically about diacylglycerols, but what do you suppose it's a lot like? Phospholipids, right? Or I was thinking even triglycerides, triacylglycerols, this is where you have 3 fatty acids. Diacylglycerols, where it's only got 2 hanging off the glycerol backbone. The third thing is either an alcohol. If it's a phospholipid, it's a phosphate, things like that. And whenever the phospholipid, or the lipase, I should say, cleaves off the second one, it becomes a monoacyl glycerol. So they call them tags, DAGs, and MAGs. But the DAG part of it is actually a secondary messenger for certain cascades. And then the one that we're going to see and talk about in greatest detail over and over and over again is called CAMP, which, what does CAMP stand for?
Cyclic AMP.
Cyclic AMP. And so it's an ATP that has lost 2 of its phosphates and has become cyclic. And I've got a picture of it coming up in just a moment. But CAMP is extraordinarily important. Where do you suppose it comes from? What's the precursor to CAMP? ATP. And so it gets dephosphorylated. And so this is CAMP. And it's, you're going to see this over and over and over again. But here's CAMP. This is a way that many times is just drawn, because there's the ribose. Of course, that's the adenine portion of it. And then it's cyclic through its own phosphate. Okay, instead of being, normally you think about the diphosphate backbone for RNA. Here, it's where the phosphate, and remember, we've talked about cyclic RNA where the 3' phosphate had been attacked by the 2' alcohol. Well, here it's very similar to that, except it's instead of 3' to 2', it's 3' to 5'. Okay, and this is the way it would look more like in 3 dimensions in that perspective drawing there. And so it's drawn a lot of times this way, but people, obviously that bond is really too large. This is where the bonds would be more drawn correctly, I should say. But this is just some of the examples of what cyclic AMP effects it can have, including, bronchodilation, inflammation involved in the inflammatory process, reducing blood pressure, the platelet aggregations, things like that. And we're going to find out, this is just a few. This is just one that I've taken from some paper that where it was talking about, specifically about forskolin, which is an activator. Very, very specific GPCR. And this is going to be, many times we're going to pick an example one, like the RTK, we'll look at insulin. For GPCR, we're talking about epinephrine or adrenaline. Okay. So what does adrenaline do, or the epinephrine do? Almost everybody uses this 3-word slogan.
Fight or flight.
What?
Fight or flight.
Fight or flight. I thought you said flight or flight. I was like, you're really flying. You're running away from there. Turn tail and run. But yes. Okay. And once again, to review, an agonist is some type of analog that will bind and elicit the effect of the natural ligand, whereas an antagonist is what binds and eliminates. So there is no response, so it blocks it. It's a competitive inhibitor. And here we're just looking at some specific examples. That's epinephrine at the top. And first of all, what amino acid does epinephrine look a lot alike? Tyrosine. We're going to find out next semester how to make it from tyrosine. And so, it's a dopaminergic. And so, yeah, it does come from tyrosine. And then we have isoproterenol, which is the agonist, and then there's propranolol, well, that one, propanolol, there we go, that sounds better, which is the antagonist. But you can see how they're very similar in structure. And then your book just shows you the actual KDs and how the agonist and antagonist actually binds considerably better than the natural ligand of epinephrine.
So would like the OH should be the one that causes the effect?
Well, it's hard to say for specific, but you can hypothesize that. Okay.
We learned to make it before finals.
Well, for next semester, yes. Your body hopefully doesn't have to learn how to make it already. It's making it. And if you want proof, I could just say pop quiz and see how quickly some of you run out the door. I do expect you to know this, the logic behind all of these, and that's one of the reasons why I did this in the past. Okay, and so this is a figure from your book, but I've broken it down just in some general spots and then put each one of them on everything. And so this is a picture. Well, this was the one for epinephrine, but it's a good generalized GPCR, which once again, GPCR stands for G protein
Receptor.
coupled receptor. Yeah, not great polymerase chain reactions. Or god-awful polymerase chain reactions or anything like that. Okay.
Sounds more accurate.
So what do you suppose, if we just think about epinephrine, which is called the beta-adrenergic receptor system, but what do you suppose has to happen first in order for the cascade to start? Don't overthink it. Epinephrine must bind. Binds. Okay. And so we have it shown here that the epinephrine comes in and has a very specific binding site. This is once again an integral protein, which to review, what is an integral protein? What makes it an integral protein? Right, it's transcending the membrane. And so this is external, so the epinephrine actually never enters into the cell, much like Moses with the Holy Land. We're going to have lots of Moses with the Holy Land moments in this class, especially even more next semester, where it can see the Holy Land, but it cannot enter in. So it binds. Something must happen in order for all of this, on the inside to occur. So in number 2, what do you suppose is happening once the epinephrine binds?
Conformational change.
You're going to have this conformational change. Okay. Now, the way that they have it worded here is, it's very, very wordy, but it's like a covert operation back there. What's happening is the epinephrine binds, it causes this conformational change that crosses the membrane, because you can imagine, binds, something happens down at the bottom, so to speak, and it's this conformational change that's going to affect what's the G protein, and the G protein actually has multiple subunits in it. Okay, and what happens here is that conformational changes causes the GDP that's bound to the G protein to be displaced. Okay, and so GDP gets kicked out and GTP is what is bound. All right, so what do you suppose is going to be happening in this third step? GDP is being kicked out for GTP. You can kind of see what's happening right there in the cartoon. So the G protein becomes activated, and what does it do? Anyone?
Phosphate.
So we do see that there's a phosphate moving somehow, but what else? What should hopefully be fairly, you can state the obvious here. What's happening right here? Whoops. Right. Or it translocates. It's going to leave that receptor to go on to the next step of the process. And what happens here is a little bit more difficult to. So in the next step, as it moves or it's leaving, is it hydrolyzes an additional GTP. So now we're talking about 2 different GTPs. The initial binding event of the epinephrine causes a conformational change. A conformational change allows for GDP on the original G protein that's bound to the receptor to be kicked out by GTP. GTP is also hydrolyzed to GDP in order for it's moving to the next enzyme, which they have abbreviated here as AC, which stands for adenylyl cyclase. And it's this binding of the G protein that activates adenylyl cyclase. Okay? Is used. And that's because the G protein itself has to be able to be recycled. Okay, otherwise you would just have one time, you'd either fight or fly. You just have a good punch in there and then that's it. And so you got to be able to regenerate this for the enzyme to be able to, for the receptor to be able to bind to another epinephrine. Okay, and so it's a little cyclic part to itself. What happens here is that the G protein has multiple subunits. It has an alpha, beta, gamma subunit. And whenever the GDP is bound, it's in the inactive state. It cannot activate the adenylyl cyclase. It can bind to the epinephrine receptor. And so what happens is, whenever you get that conformational change, let me make sure I don't move it, GTP kicks out the GDP. It activates the alpha subunit, and that's what this is now showing. This alpha subunit can then move on to go to adenylyl cyclase. The beta gamma subunits are then recycled to go back to await for the reemergence, or for the return, I should say, of the alpha subunit.
So it can bind to the AC, the adenylyl cyclase, when GTP is on it. So why does it hydrolyzes just to turn itself off?
Yes.
So the hydrolyzer doesn't do anything like work, really, just deactivates it.
No, I mean, the hydrolysis we're going to find out is what's going to, it's going to bind to adenylyl cyclase. It's going to activate the adenylyl cyclase through this when it's hydrolyzed, so that way it can only do it one time, and then it goes back, or it can go back, I should say. And so that's why eventually an inorganic phosphate will be kicked off. Then this GDP-bound alpha subunit can go back to the receptor, bind to its beta-gamma subunits to await the next epinephrine to come. Okay? And one of the reasons why this is very important is, let me see if I can blow this up so we can see it better. Ooh, I can. Is that cholera, this is where cholera works. Okay, and so normally we have, this is normal GTP, it's got what's called normal G protein's got GTPase activity, which is what ends the signal for adenylyl cyclase. Okay, so as long as this GTP is bound there, it's this adenylyl cyclase is kicking, it's turned on. Okay, once it gets hydrolyzed by its own GTPase activity, then it's going to be able to leave, and the adenylyl cyclase is no longer turned on. What cholera does, and this is just showing how this works. What cholera does is the cholera toxin then modifies it by adding, on an important arginine, it adds this ADP ribose subunit from NAD. Once it's ribosylated, it's constantly turned on for the adenylyl cyclase. So that's really important to know. What happens when you have cholera? When you're suffering from cholera?
Fluids everywhere.
So, in what ways?
Diarrhea.
Doesn't it create holes in your intestines with fluid?
Well, I mean, it's known for its intense diarrhea. Like, it's just, like, you cannot, it messes with your electrolytes because you're constantly going, and it's, yeah. So, I mean, that's just one of the phenotypes of it, but it does lots of things because it's constantly turns on the adenylyl cyclase. So adenylyl cyclase is continually doing its business while causing you to do your business. Be here all week. Well, not really, because next week is Thanksgiving. So enjoy me while you can. But yeah, so this is like the effect of the cholera. What do you suppose, and I know it says, oh, enzyme, and the funny thing was, which by the way, that answer is adenylyl cyclase. I had people miss it. I'm like, it literally, not only does it say AC, but right next to it, it says adenylyl cyclase. But people would miss it before. All right, so we have adenylyl cyclase, and it takes an ATP, and what do you suppose it's going to do? Listen to the name, adenylyl cyclase. So cyclases are enzymes that do what? Make something cyclic. I guess if it works in reverse, it could, take a cyclic thing and break it. But yes, it makes things cyclic. And in particular, what do you suppose like adenylyl cyclase is going to make cyclic? It's going to make cyclic AMP.
Oh, yeah.
Oh, yes. What are we making ultimately?
Cyclic AMP.
Okay. And so it starts with this compound. What's the first compound there? That is ATP. Okay. So we have ATP. We have an enzyme that's going to take ATP. It kicks off, the PPI is inorganic pyrophosphate. It's two phosphates. It's a diphosphate that gets kicked off to make the cyclic AMP. Okay. All right. So All right, so here we have cyclic AMP, and cyclic AMP has to, it goes on and does its various things, but we have to be able to get rid of the cyclic AMP also, or otherwise you'd have the same effect as you would with cholera. Okay, that secondary messenger is going to go on and do its good stuff, but if there's only so much, you have to pay the piper if you continue to have the good stuff done, okay? So you have to have a way to turn off the signal. And so that's what the second enzyme is doing in the metabolic pathway of cyclic AMP. And so we have water coming in, and water is breaking apart that cyclic bond. So what compound are we making right there? That's AMP. It just didn't seem monophosphate. So what enzyme do you suppose is doing this? What kind of enzyme or class of enzymes could this be?
Hydrolase.
A hydrolase? Possibly. What else? What other kinds of enzymes are there? There are hydrolases. What else? Pardon?
The lyase.
Maybe a lyase, especially if it went through some type of intermediate that had a double bond. What other classes of enzymes are there? Oh my gosh. I mean, one day we're going to have someone from psychology fall through the floor there through our roof. What other classes of enzymes? You can always go through, could this possibly be a, it can't be a protease because this isn't a protein that's being degraded. Could it be a kinase? No, because the phosphate's not being transferred using ATP or GTP. What about like a hydratase?
Sounds good.
Possibly. What about like an oxidoreductase? No. Usually those are ones that uses NAD, NADH, FAD, FADH2, where you're losing or gaining electrons. So we have our promiscuous enzymes, because cyclic nucleotides is what it works on. It's breaking down a phosphodiester bond. Okay, so it's taking CAMP, turning it into AMP. And whenever we need to get rid of the CAMP will keep going on and on and on and going to what's on the right-hand side of this figure until the cyclic nucleotide phosphodiesterase hydrolyzes that bond, okay? And then it becomes AMP. We'll find out AMP is important later on. It's another messenger in and of itself. AMP can be used to make ADP or ATP as needed. But CAMP itself goes on, and I'm just going to have to tell you what it's going to do. CAMP is going to turn on another enzyme. CAMP is a secondary messenger. It's going to go turn on that third tier enzyme, and this enzyme is what's so important. It's called protein kinase A, which they abbreviate as PKA. There are lots of PKA has lots and lots and lots of uses and other enzymes and processes that it affects. It's the one that, does a whole host of things. But it's a protein kinase, and then the A is because it's turned on by cyclic AMP. And kinases, what do kinases do? They add, or they could, in theory, if they work backwards, remove the phosphates using ATP or GTP. Here we're using ATP, okay? And so if it's a protein kinase A, that also tells you that's promiscuous. It's going to be phosphorylating lots of other different proteins at the expense of ATPs. Okay, so that's what that means by what's happening and everything. And then, like I said, there's just lots and lots of things that this enzyme. And so we have that CAMP activates PKA. PKA is what goes on to cause eventually all these cellular responses to that initial binding of epinephrine. Because there's lots and lots and lots of proteins that can become phosphorylated. It's going to turn some of them on, and it's going to turn some things down, regulate other things. Because phosphorylation doesn't always turn everything on. There are times that phosphorylation will downregulate it will downregulate systems. Okay. Are there any questions over this?